Hello students, I am welcoming you all for the today's class. I hope you are all fine. So in the previous class we have discussed about the six kingdom system of classification. In this class we are going to deal with the concept of kingdom Monera. So before starting this concept I want to tell you one thing. Subscribe for my YouTube channel and as well as join my WhatsApp channel. The link has been given in the description. So for all the biological updates, join my WhatsApp channel also. So now let's move to the concept of Kingdom Monera. Kingdom Monera. Bacteria are the sole members of Kingdom Monera. As you know, bacteria are the members of Kingdom Monera. Bacteria are placed under the Kingdom Monera, so they are the sole members of the Kingdom Monera. Bacteria are present everywhere. As you may see the bacteria everywhere. Inside the human beings also some of the bacteria can live. The example for bacteria are E. coli is a general bacteria that is living in the small intestine of an human being. So the bacteria are living everywhere. So billions of bacteria are present in handful of soil. If you take a handful of soil, then you can observe billions of bacteria. Not millions, billions of bacteria are present in handful of soil. So I hope you have cleared the second point also. Bacteria are present everywhere. In the handful of soil, billions of bacteria are present. Bacteria can live in extreme harsh habitats such as hot springs, desert, snow, and deep oceans. Bacteria are the organisms which can live in extreme harsh habitats, means extreme high salty areas are they can survive, as well as in high temperature areas also they can survive, as well as in deep oceans, and in the snow also bacteria can survive. Bacteria can survive in hot springs, desert, snow, and deep oceans. Bacteria can stay everywhere and can live everywhere. So bacteria can live in or on the other organisms as parasites. Bacteria they can be live in inside the organism body, or bacteria can live on the other organisms as a parasites. As a parasites, bacteria can live on the other organism because you know bacteria, the majority of the bacteria are heterotrophs. Heterotrophs are nothing but they can't prepare their own food. They always depend on the other organism for the sake of food shelter. So bacteria can live in independently, or they can live on the other organism as a parasites for the sake of food. I hope you have cleared the third point also. Some of the bacteria are autotrophic. As you can see, some of the bacteria are autotrophic. I am not telling you all the bacteria are autotrophic. Only few of the bacteria are autotrophic. Autotrophic. As you know, autotrophic is nothing but the organism which can prepare their own food through the synthesis of inorganic substrate. They can prepare their own food. Very few of the bacteria are coming under the autotrophic category. That autotrophic organism that may be photo or chemoautotrophic. The bacteria, very few of the bacteria are autotrophic. Under autotrophic, they may be photoautotrophic or chemoautotrophic. Photoautotrophic means through the sunlight they can synthesize their food. So that is photoautotrophic. And chemoautotrophic is nothing but through the synthesis of chemicals, or with the help of several types of chemicals they can prepare their own food, or through the several chemical reaction they can prepare their own food. They are generally termed as chemoautotrophs. But the vast majority of the bacteria are heterotrophic. The vast majority of the bacteria are heterotrophic. Very few of the bacteria are autotrophic, but the majority of the bacteria that possesses the heterotrophic character. Heterotrophic means they always depend on the other organism for the sake of food. They can't prepare their own food, so they are heterotrophs. The majority of the bacteria are heterotrophs. Hetero means other, trophic means mode. The organism which depend on the other organism for the sake of food, they are generally termed as heterotrophic. Majority of the bacteria are heterotrophs. Okay, I thought you have cleared the this point also. Kingdom Monera is divided into two groups. The kingdom Moneria is further divided into two groups. That two groups are, one is Archaebacteria and another one is Eubacteria. Archaebacteria is nothing but they are primitive bacteria. Primitive means they are very small. You can't see by the naked eyes. So you have to use the microscope to see their structure. So the first one, Archaebacteria, is the primitive type of bacteria, and Eubacteria is the true bacteria. True bacteria is nothing but they are having proper cell wall. True nucleus is present. So they are termed as Eubacteria. Okay. I thought that you have cleared about the concept of kingdom Monera. Now moving to the first group of the kingdom Monera, that is Archaebacteria. The first group of the kingdom Monera is Archaebacteria. As you know, Archaebacteria are the special type of bacteria that can live in the habitats such as hot springs and in high salty areas, and as well as in the marshy areas. You are seeing here, these are the special type of Archaebacteria are the special type of bacteria that can live in high salty areas. The bacteria that can live in high salty areas are generally known as halophiles, and the bacteria that can live in hot springs, they are known as thermoacidophiles. As the name itself indicates, thermo means heat or a high temperature. The bacteria that can live in high temperature areas, that are generally termed as thermoacidophiles, and in the marshy areas, the bacteria that can live in marshy areas, they are generally known as methanogens. Archaebacteria are the special type of bacteria that can live in high salty areas. Now moving to the second point, these bacteria differ from other bacteria. What happens is Archaebacteria differs from one bacteria to another bacteria in having a different cell wall structure. The bacteria to bacteria, the cell wall structure will differ. So the cell wall structure will differ. Why this happens means it is required for the survival in extreme conditions. This feature is responsible for survival in extreme conditions. So that reason the bacteria to bacteria cell wall structure will differ. We are seeing here this bacteria differs from other bacteria in having different cell wall structure, and this feature is responsible for survival in extreme conditions. Extreme means harsh conditions. So that is the reason why the bacteria are having different cell wall structure. So I hope you have cleared that point also. Methanogens present in the gut of several ruminant organisms such as cow and buffalo. Cow and buffalo and other organisms are there in their guts. They are having methanogens. Methanogens are present in the gut of several ruminant organisms. The example for ruminant animals is cow and buffalo. These are the animals which are having methanogens present in their guts, and that methanogens are responsible for the production of methane. For the production of methane, these gut of several ruminant organisms are required, where there is a production of methane will take place. Biogas from the dung of these animals. From the biogas is created from the dung of these animals. and that will lead to the production of methane. Methanogens. Some of the examples have been given here. In case of methanogens are nothing but the bacteria that are living in a marshy areas that are generally termed as methanogens. The example for methanogens is Methanobacterium, Methanobacillus, Methanococcus, Methanosarcina. As you are seeing here, Methanobacillus. Bacillus is nothing but rod-shaped bacteria that are generally known as bacilli. So these are rod-shaped bacteria. Coccus, Methanococcus. Coccus are nothing but they are round-shaped bacteria that are generally termed as Methanococcus. Coming to the next point, halopiles. As you know, halopiles are nothing but the organisms that are living in high salty areas that are generally termed as halopiles. The example for halopiles is Halobacterium. Halobacterium is the example for halopiles and Halococcus. Halococcus is also an example for halopiles. Coming to the last point, that is thermoacidopiles. As I have just now told that the bacteria that are living in hot areas or under the hot temperature or living in high temperature, they are known as thermoacidopiles. The example for thermoacidopiles is Thermoplasma, Sulfobolus, and thermoproteins. These are the three types of bacteria that comes under the category of thermoacidopiles. I hope you have understand the first group of Monera, that is Archaebacteria. Okay. Now moving to the second group of kingdom Monera, that is Eubacteria. Eubacteria. Anton van Leeuwenhoek is the one who discovered the bacteria and he called them as worms. Anton van Leeuwenhoek, in the year of 1675, he discovered the bacteria and he called them as worms. In the year of 1675, Anton van Leeuwenhoek discovered the bacteria and he called them as worms. Traditionally, bacteria were believed to be, in the traditional, the bacteria were believed to be unicellular microscopic plants. Traditionally, bacteria were believed to be unicellular microscopic plants. As you know, unicellular means uni means the organism that is composed of single cell, and microscopic means micro means small sized organism which cannot observe by our naked eyes. So plants, as you know, plants. So traditionally, bacteria were believed to be unicellular microscopic plants without the chlorophyll. Without the chlorophyll, in terms of plants, the chlorophyll is present. So, without the chlorophyll and reproduced by binary fission. Traditionally, the bacteria were believed to be unicellular microscopic plants without the chlorophyll and that they reproduce mainly by binary fission. Binary fission, as you know, the single parent cell that divides into two daughter cells after some after few days, each daughter cell will develop into an identical parent cell similar to the parent cell. Similar to their parent cell, each identical daughter cell will generate. So that is binary fission. Bacteria, Ehrenberg first of all coined the word bacteria. Ehrenberg is the name of the bacteriologist who first of all coined the term or the word bacteria. Bacteria divided into four groups. The bacteria that are divided into four groups based on their structure and shape, the bacteria are going to be divided into four groups that are namely round, round shaped. The round shaped bacteria that are known as coccus bacteria or cocci, and rod shaped bacteria that are generally known as bacillus or bacilli. The bacteria which is in rod shape, that is generally known as a bacillus bacteria. If it is a round type of bacteria, then it is coccus bacteria. If it is rod type of bacteria, then it is bacillus bacteria. And the bacteria which is having comma shape like this, then it is called as Vibrio. The bacteria which is having comma shape, then it is known as an Vibrio. The bacteria which is having spiral shape, spiral is like this. The bacteria which is having a spiral shape, then it is known as an Spirillum or Spirilia. I hope you have cleared about the different types of bacteria based on their shape or structure. Bacteria that have significant appear on human life. The bacteria that play a significant impact on human life also. You can ask me how bacteria play a significant role in the human affair. So, in the production of curd from the milk, several types of bacteria are involved. For example, lactobacillus is the bacteria that is involved in preparation of curd from the milk. Through the help of the bacteria, only the curd will prepare from the milk. So, bacteria plays a significant impact on human affairs as well as in the production of antibiotics. Also, the bacteria plays a major role. Like penicillium is used in the preparation of antibiotics as well as in fixing the nitrogen in legume roots. In bacteria plays a very important role in fixing a nitrogen in legume roots such as cyanobacteria. Cyanobacteria is the best example that can fix atmospheric nitrogen in the legume roots, in the legume plants like in groundnut. So the bacteria have significant impact on human affairs. Different pathogenic bacteria cause human diseases. See, bacteria are beneficial also and bacteria is harmful also. Some of the bacteria causes damage to human beings as well as forms as well as animals as well as pets also. Some of the bacteria can cause damages. So different types of pathogenic bacteria. The different types of pathogenic bacteria. Pathogenic is nothing but disease transmission, or it is a pathogenic means it cause damage to the life. The different types of pathogenic bacteria causes human diseases like cholera, typhoid, and tetanus. The cholera, typhoid, and the tetanus are the diseases caused by the pathogenic bacteria. As you know, typhoid is caused by Salmonella typhi is a type of bacteria. Through that bacteria only typhoid will occur. How typhoid will come? Typhoid will come to the human beings by drinking contaminated water or by eating the contaminated food. So the human will going to have the typhoid. So Salmonella typhi is the bacteria that is involved and causes typhoid disease. So, like this manner, the pathogenic bacteria causes human diseases. Not only human diseases it will also cause the plant diseases also. Bacteria can also cause the plant diseases also. The plant diseases like citrus canker. As the name itself indicates, citrus means lemon, orange. Several types of fruits will come under citrus. So, citrus canker, as you see, it is a suppose it is a lemon. On the surface of lemon, you are seeing a black color dots like patchy black color dots that are nothing but citrus canker. Means that fruit is suffering from or it is having citrus canker disease. And blight of paddy. Blight of paddy is also a disease that is caused by bacteria in the plant. And other example is crown gall of apple. Crown gall of apple is also caused by the pathogenic bacteria. I hope you have cleared this point also. Now moving to the reproduction. how the eubacteria will reproduce. So now let's discuss. The eubacteria reproduce mainly by binary fission. The eubacteria that reproduces mainly by binary fission. Just now I have explained that the parent cell that divides into two identical daughter cells. Both of the daughter cells will develop similar to their parent cell. That is nothing but binary fission. The eubacteria mainly reproduces by binary fission, and in the unfavorable conditions, whenever the conditions are unfavorable, that times this eubacteria produces the endospores, and that endospores are the bacillus species. That endospores are bacillus species. As you know, bacillus means rod-shaped type of bacteria. Okay, reproduce mainly by binary fission. Under unfavorable conditions, it produces endospores. Endospores are nothing but they are bacillus species. They also reproduce by sort of sexual method of reproduction also. Eubacteria also reproduces through sexual method also. You can ask me how eubacteria can reproduce sexually. By adopting a primitive type of DNA transfer. Primitive means very small, which you cannot see by naked eyes. So by adopting a primitive type of DNA transfer, as you know, DNA is a genetic material in the bacteria. So by adopting a primitive type of DNA transfer from one bacteria to other bacteria, this is what sexual reproduction will occur. In this way, sexual reproduction occur in the eubacteria. The several steps involved in the sexual reproduction are conjugation. Conjugation is nothing but transfer of genetic material, that may be RNA or DNA. Transfer of genetic material from one bacteria to another bacteria is generally termed as an conjugation. The several steps that are involved in sexual reproduction are conjugation, transformation, and transduction. These are the three steps that are very important for the sexual reproduction in the eubacteria. So from the one bacteria to another bacteria. So I hope you have cleared the two different groups of kingdom Monera. First one is Archaebacteria we have discussed, and the second one is Eubacteria. Now we have discussed. In this class we have discussed about the kingdom Monera, characteristics of the kingdom Monera, as well as two groups of kingdom Monera. One is Archaebacteria and the second one is Eubacteria. I hope you have cleared these all concepts. Under kingdom Monera, we have left with the three concepts that is cyanobacteria as well as mycoplasma, actinomycetes. These three uh, concepts are left behind. So in the upcoming classes, in the next class, I will discuss that three concepts also. Subscribe for my YouTube channel. Thank you one and all. Thank you. Meet you in the upcoming classes.